like this guy that, Kalyan. He always keeps his formal. That's why he asked me to say something. Nothing to say because we are all friends. Number one, we are really happy to say that this group, because we have got 7 working groups. Out of that, this working group is the most active group. So this is we actually applaud their activity. Of course, there are members who and they always join in this type of activities, and the leadership also are also very active. As Kalyan was very generous to give me 5 minutes, so I have to take my 5 minutes. So it is just plasma sourcing and fractionation scenario in our member countries. We are actually blessed. AATM is blessed that we have got 23 member countries from 4 WHO region. It is the Sierra, Wipro, the Asia Pacific, AMRO, Middle East, as well as Euro. We have got 4 member countries from the Euro, and I request all members who have not yet registered, they should register for our forthcoming meeting, where Sangeeta is our scientific president or coordinator, something like that. I don't remember all these designations. So
yes,
yeah, you join, and this will be, I can say that you will not find any lack of scientific topic. It is actually 3 parallel halls. And in one hall, we are going to discuss about fractionation, and this will be organized by this group. Secondly, talking about the fractionation scenario in our in this region, that actually this is a greater Asian region. We work from Turkey to Indonesia, as well as from Media, and where we work. And in this area, there are lots of work to be done in fractionation, because most of the countries and are actually discarding plasma. This is actually our responsibility to get it, some sorts of responsibility by this group take for proper plasma sourcing. However, there's an interesting figure, that there is not any shortage of fractionator, and you'll be surprised to know that there are 6 fractionators at this moment in India. So, of course, our collection is more. It is almost 13 to 14, 13 million units at this moment. And as per government statistics, almost 75 to 80% are prepared components. So 4 fractionators, so there should not be any problem for our member countries. However, this is just a background information, and just to put some thoughts in your mind, how we can help our this without going further. I request Kalyan to take it forward, the scientific part of this webinar. Thank you very much to everybody. So giving a patient hearing to my, whether you call it Bhagwas or whatever it is, but it is the, opening one. Thank you very much. Thank you.
Kalyan, you're mute. Kalyan, mute.
Okay, sorry. Thank you, Navjot sir, for your nice words and encouraging me. And I would like to request our chairperson of the Atom Plasma Protein Therapy Working Group, Dr. Sangeeta ma'am, to say some words. Dr. Sangeeta,
very good to all of you. And we are actually very delighted to have Professor Hari Nair with us today, and who will be speaking on self-sufficiency in plasma-derived medicinal products for the emerging nations, which is a dream or a reality. And why a dream or reality? Because we all know that human blood is the source of these multiple therapeutic products, which includes the PDMPs and the blood products. are fractionators and the people who are working with plasma. Of course, it's not a new term. And we need to, on this side of the table, we all need to learn so much about it. And there is always a shortage of the plasma. Like in India, there is a sufficient, but how much of it actually, that is a problem, and that is a question. That is what Dr. Nair would be talking about. And though I have been given 5 minutes, but I'll hand over to Kalyan again, welcoming you all once again. And we cannot wait to hear from Dr. Nair. So I'll hand over to.
Thank you, ma'am. Thank you for your encouraging words. And now I'd like to request Dr. Ranjit Ajmani, sir, to formally introduce Dr. Hari Nair, sir. And the floor is yours, Dr. Ranjit.
Thank you, Kalyan, and thank you, Sangeeta and Navjot. I think it's absolute pleasure for me to introduce Hari on this forum. Hari and we have been friends for quite a while. We have traveled the journey of plasma fractionation, plasma, and so on and so forth for a while, almost more than a decade now. And this journey has not been very smooth. It is bumpy ride all through. And probably it would remain for a while till the time we come to a state of equilibrium or settlement. And the challenges are humongous. If you look from the plasma, is plasma available or not available? And if it is available, is it safe? So there are multiple questions. So Hari, from that background, being a trained hematologist, but in the coagulation factor treatment, he has done quite a pioneer work, and he's also professor in on almost 30 plus years of work has gone into this developing a new technology. And whenever a new technology comes, it comes with apprehension, whether it is no one would have believed that we will be holding a mobile in a hand and talking to people. It was a distant dream maybe 30, 40 years ago. So everyone enjoys the status quo. Any change, any disruption brings a lot of discomfort. So, and it comes with a legit help, in many ways, not just in the sense of, the setting up a fractionation plant, archaeology. It's a quite holistic view we are looking at it. And this is the way I think Hari will take this discussion forward. And I hope, Hari, this introduction will take you to the next step, and the floor is yours now.
All right. Firstly, I'd like to thank everyone for allowing me this opportunity to talk to you, especially the organizing committee for listening to me. I have to tell you that I'm going to be a little controversial today, and I want to be controversial so that I can get people to think about what it is that we require in the Asian region, and also where emerging countries are concerned. I love the topic. I have to tell you, the way that it was coined and the way that Ranjit approached me with this, where you're asking whether it's a dream or a reality. Ranjit and I have been friends for a very long while, and he's now joined my company as the CEO for AGROS Asia. And I think that what we are trying to do here is to help people. At the end of the day, fractionators and everyone else, they're business people who want to make money. At the same time, if we make money, but we also help people, that's what we should be all dreaming about. And as a doctor, I think that helping people comes first. And that's not exactly what my financial people would want to hear most times. Now, what are PDMPs? There are 23, 25 PDMPs, and you can see this list. And for those who don't know what it is, it stretches from all the coagulation factors to immunoglobulins, to albumin, to fibrinogen. So there are a number of these things.
But you know what? There are 3,000 proteins in plasma. And in clinical practice, we only use about 25, which tells me that there's a lot to be mined. My business partner, John, who's actually on this call, talks about mining the plasma. And mining the plasma means that you've got various other proteins that you can actually use in a clinical setting. And in many instances, we don't even know what they are. So what is the world situation? $28 billion, that's the therapeutic market. And of that $22 billion worth of product, 20 million liters of blood plasma gets exported from the United States. Interestingly, that goes everywhere. Now, the day the United States turns off their plasma supply, this will impact everyone. And it'll impact many countries in the world, especially the emerging countries and especially Asia as well. So we have a duty, as Sangeeta was actually saying, to make sure that people in Asia, the immunoglobulin demand is growing at 7% per annum. Now, this is versus a plasma supply growth of 4%. Now, you know, it's pretty obvious that demand is outstripping supply. And in countries like Asia, there are many indications that we do not even treat because of the fact that there is no availability of the PDMPs that are required to treat these indications. So increasing process yield, in my view, is the only solution. The current highlights are this, right? Approximately 55 million liters of plasma is fractionated annually globally, right? That's a lot of plasma. 70 fractionation facilities. In fact, there may be less now because there have been many mergers and acquisitions. And the main technology is 90 years old, cold ethanol precipitation plus modern chromatography. Nothing new has come about in this time period. And where things are lacking is that the current technology at best gives you about 50% yield. And I'm talking about process yield. And then, of course, we've got two orthogonal pathogen inactivation steps, prion removal, and then dealing with new viruses, especially in the Asian type scenario where you're looking at things like Zika and chikungunya, and you know, God knows what else. The interesting element of all of this is that the more steps you add to the process, the more losses you have as well. And that at the end of the day, the process yields cannot go any further than 50% unless you're looking at new technologies. So in the emerging economy scenario, what are the challenges? So we know that WHO has come out and talked about a mandate which says that self-sufficiency is the way to go. Now, what does that mean? Does that mean that every country in the world must set up a local plasma fractionation plant? You know, by golly, that's an expensive, complex situation. And it is something that we need to consider when we talk about self-sufficiency. Does self-sufficiency mean regional joint ventures? And then, of course, it comes down to the question of whether governments can actually work together so we can have regional joint ventures. Then you ask the question, contract fractionation. So let's send plasma to established fractionators. The problem is, and this is the reality, you always become second-class citizens to the major fractionators who are centered in first world countries. And so, some of that means that buying commercially means that you get a dumping of plasma proteins or PDMPs. And when I say dumping, I mean, you know, the products have a very short shelf life. So that's not something that you can even think about. And of course, the age-old thing is, well, let's try pathogen inactivation of FFP and cryoprecipitate. Well, I think, and I'm quite happy to be quoted on this, that's a second-rate solution to a first-class problem, all right? Because it's not a question of, to me, ever since I was a little boy, I've always said that medicine is not a privilege, you know, it is a human right. And that means that everyone deserves the best possible medical care if we can afford that to happen. So plasma fractionation in emerging nations has its origins in, plasma fractionation in general has its origin in medical service industry. It then evolved into a global manufacturing industry. The thing is, it's a highly sophisticated industrial process, and it has become the most probably sophisticated industrial process that we have in the production of biological goods. It's regulated, and the highly regulated part of this comes because of the requirement of high standards of product safety. And this has come about because of the fact that bloodborne diseases happen to be present in human blood, in human plasma, and things like HIV. When we had the scares of HIV in plasma, when you're looking at things like factor VIII, when we had mad cow disease situations. So the regulators have become very, very strict to ensure that the final product that you give a patient is safe. And it's a rigid confirmation to GMP, good manufacturing practice, which ensures that you do that. And then, there are a number of things here which is a big challenge when you're looking at an emerging economy. And then, as my friend Ranjit has always pointed out, the dependency on a stable electrical grid. Why? Well, how do you store plasma at minus 20 degrees if you can't rely on electricity? It just doesn't work like that. Then there's this major thing, the availability of raw material. I just heard, the business about 30 million liters of plasma in India. And the question that Dr. Sangeeta has asked is actually very, very important. What is the usable plasma volume in countries like India? And forget about India, because India is a growing economy. In the smaller countries, the volume sweet spot really, in my view, is about 30,000 liters to 50,000 liters per annum. Using current technology, you cannot process, and we have to look at the commercial side of things from an economical profit and loss scenario, process 30 to 50,000 liters using current technology and become viable and successful. It just does not happen. So we're talking about in terms of the requirements of plasma. The number of blood collections will increase in emerging nations, and the reason for that is blood components, your red cells, white cells, etc. These are hemorrhage and trauma, and all you're doing now is generating more and more plasma. The volume of wasted plasma, and I use the term currently unusable plasma cup, it's wasted plasma, was estimated. Now it's estimated to be around 9.3 million. So you're really talking about large volumes of plasma that literally are poured down the drain, and that is, in my view, criminal. Now, the collected plasma in emerging economies, what we collect normally, is considered not fit for fractionation by current fractionators because of the regulatory requirements of actually having donations which are at the FDA or EMA level. Now, this is a big challenge, because you've got to ask the question, and I keep asking this, whether it is justifiable, whether it's a dream or a reality to think that all plasma that's collected everywhere in the world will be at the same high quality level that you expect in first world countries. Now, the other aspect of all this is, 60% of the population of the world reside in Asia. And it is such a no-brainer to look at this and say, well, Asia is a critical partner, and I use the word critical partner, in global sufficiency of plasma, as plasma becomes a difficult resource to supply. Now, if you look at the plasma dynamics in sophisticated fractionators, the yield are the two things that drive whether fractionators can become successful or not. And this is a big, big problem when you're looking at new fractionation plants. So the big thing, all right, you have a plasma fractionation plant, you need to establish a regulatory body in the country, which in itself is expensive to maintain. Now, on top of that, you've got to have this regulatory body, which then is capable of policing the fractionators. On top of that, you need highly trained technical individuals. Is this even possible? You got to ask the question, don't you? I mean, is this even possible? And the quality of the plasma, well, it doesn't meet the quality required for current fractionators. So what got to upgrade blood collection organizations so that you collect plasma at the level required by fractionators. I ask the question, is this even possible? In the 80s, the regulators said, well, the blood collection companies need to do proper screening, testing, and then have the ability to trace back donors and also companies where these things were collected. So we started using many techniques. We started using viral inactivation steps, and all companies, and I mean all companies, are required to validate what they do. So you went from pasteurization, dry heat, solvent detergent, etc., all the way to initial screening. Because you had epidemiological surveillance, you had donor screening, you had donor testing. So there was a whole period that you had to look at. You had mini pool virus marker testing. That meant that things like solvent detergent, for example, was also introduced. Why am I saying this? Because if you look at the evolution of viral screening and viral reduction, you start in 1984 with dry heat. You then go to 87 with solvent detergent. 1990, it suddenly turns to dry heat, but at a higher level. HIV, all envelope viruses, HAV, B19, all envelope and non-envelope viruses. So the combination was that at the end of the day, you had a very, very safe product. But have a look at what all this means. It means that at the end of the day, the actual process is also very, very expensive and very, very difficult to police. The question that we have to ask is, what can be changed? All right, so the simple answer is reduce validation requirements when the product is processed using the same conditions, albumin, IgG. And I think that there's a case to be made for this, because when you look at validation requirements, there is an overkill at the moment. And the overkill is so that we can make sure that it is safe. So with controls and testing that are in place for each donation, do companies still need multiple methods of viral inactivation and removal? And I think that the answer is no. But it requires regulators to think actually outside the box and also think about what it is that we can do within modern medicine and modern science that allows you to do things differently. This whole concept of artificial intelligence, you know, the introduction of this, we should be able to better manage risks as we start looking at revolutionizing plasma fractionation. Go to the next one. So the question that you have to ask then is, can every emerging nation be a fractionator? And my humble view, and it is a humble view, is that I don't believe that that's possible. All right. And the reason for that is that if you look at fractionation, it is very difficult and very expensive for every nation to have a plasma fractionation plant. But can you look at a hub and spokes model as a better alternative? In other words, you look at countries where you can actually introduce plasma fractionation, where you can actually look at regulations. regulatory body, and then start looking at these fractionators importing, and I'm talking about in your own region, importing plasma that allows you then to get a supply of products that can be used in these countries. Then the question becomes, is this even fundamentally possible? Because can you actually have a centralized regulatory body which can monitor, liaise, work with emerging country governments? Can we use, for example, an AATM-like association, like the one that I'm addressing at the moment? Can AATM actually be a starting point where we can have a EMA type a body that can look at regulation in all these countries so that it can be somehow harmonized? I am not even suggesting to you that this is a simple move, but again, you need to start thinking outside the box. Can we manufacture a protein with less stringent plasma collection quality, which still meets with Western regulatory standards of production, quality? Then only a dream. And I don't believe in dreams. I actually believe in challenges that we can actually meet. I have no idea why my screen for this change to use less qualified plasma, but manufactures a product which still meets the most stringent regulatory standards. And I would like to prefer that tangential flow electroseparation, which is an invention that I'm very proud to say I'm a co-inventor of, is just this. And I want to give you that my vision of how this is possible within an Asian type context. So the hemofrac, which is this form that you see at the back, is something that we use in Australia for the fractionation of hyperimmunes. We are in the process of building a 1 million liter plant in Brisbane, and we're building a plant in Singapore, which is also a 1 million liter plant. And so the Singapore plant, countries here, including Cambodia, we hope countries like Laos and Myanmar, etc., where they would be able to send us the plasma so that the plasma can then be manufactured in this particular scenario. So what is this? All right. If you look at the technology is composed of 3 membranes in a cartridge. All right. And the separation membrane in the middle allows you to have different pore sizes. The molecules, the proteins themselves, can be charged, and the protein can be charged depending on the buffer that the protein actually floats in. So you've got plasma, you've got a buffer, and say if the buffer has a pH above or below its isoelectric point, you charge the protein. And then in an electrical environment, out of the 3,000 with very high purity and very high yield. Currently, you're talking about yields of about 50%, as I said, with look at what we're doing, you're looking at process yields of about 90% and over. So in effect, 1 liter of plasma, you get double the protein that you need. Now, that capable of producing more protein, and therefore there's a greater supply to meet the demand. But can you do that safely? And that is the question that I think we need to question today. Oops, okay. So this hemofrac system actually consists now, the hemofrac system actually consists of disposable membranes. And if you compare this with chromatography or cone ethanol precipitation, you're looking at solid fixed bed systems, you're looking at the use of ethanol. None of use all those sort of things to actually also destroy the protein that you're targeting. So this particular invention stands all that, and you get very high quality protein. Now, what you see from this chart, from the CSL right down to the LFB columns, is something that MRB, the Market Research Bureau, has put forward. The mean yield here we're looking at is about 3.87, and this is for immunoglobulin. For our product, which is the one in blue right at the top, you're looking at an average of 7. So you can virtually see that what you're doing is you're doubling the yield because you've got a new technology. Now, if this new technology is to work, it needs to also remove viruses. And what we have done is we've had a look at a number of studies which actually show that this is actually the case. In normal situations, what happens is that you have nanofiltration, right? So that membrane that you see in the center is a nanofiltration membrane. Nanofiltration works on a system pressure. You push through the liquid through that membrane, and then what will happen is your IVIG comes out. But because of the high pressure, there is a possibility and that you also will get the virus through. And that is something. So pressure forces viruses through the nanofiltration, which can then cause contamination. In our system, the thing is very different. We have a system where the viruses are usually negatively charged. And again, this is because of the buffer that we are using. And if you use a negative of the buffer, the only thing that comes out is the IVIG, because the viruses themselves cannot come through because of the pore size of the membrane, but also because they're negatively charged, they're held back because of the positive electrode that you have out there. So that's two forces which work in tandem. Viruses that we've had a look at. So we've had a look at anything from B19 and envelope viruses of many different sizes. And of course, the question is, well, if you've got envelope viruses, then what about the non-envelope viruses? Well, if you look at this particular slide, we used viral models which are non-envelope and BVDV, which is the other one that we. And what we found is that in each case, the IG product, there is a dramatic decrease in the viruses greater than a 6-log reduction. Whether you use a nested PCR or an infectivity assay. The number of products that we've looked at, albumin, alpha-1, PI, IgG, hemoglobin, all of which one primary step. It's a capture step which does this, and reduction steps. So literally, you're looking at say high. The next slide. We've dreamt about, but have a look at the purity of the product. This is fine that there is no virus that comes through if it's loaded in the alpha. Have a look at the IgG purity of the product. Again, you don't. So essentially, whether it's an enveloped or non-enveloped virus, you're getting dramatic decreases in the envelope viruses. You're actually getting a complete reduction. And even here, this is what you need in terms of viruses, we get the same sort of results. Now, this fusion service, and we looked at IgG separation. paper greater than 3. The only reason for that is the time that we did it, the spectacular results. And in fact, in many of the actual pictures that we were seeing, we couldn't see any prions whatsoever. So you have got a gap, all right? The raw material that you are getting not necessarily be collected at the level that you're talking about because of issues of upgrade, because of the technology. So plasma, that's a huge challenge. But again, the technology MP program needs to be policed. The availability of skilled personnel. And, this is one thing that most people requires highly skilled individuals. And there has to be a relationship between plasma fractionators and university, the standards of requirements to actually practice in these plasma fractionation structure and organization, and then the harmonization of regulatory requirements in the region. But the thing that I want to put across to you here today is this last point. Not countries, but the question is, do we, as a group, do we in technologies of the past, but we need to be brave enough to use this. The final product has got to be of the highest quality and of the highest safety. That is never, ever to be compromised. But the regional focus on self-sufficiency should be a collective project and not necessarily country-specific, because I don't think that you can make it happen country-specific in emerging nations. So the question that was posed when and positive steps in a collective collegial manner to actually succeed in this process of making sure that Asia comes. Rajit, are you there? Can you hear me?
I'm pretty much around. So thank you very, very much for giving a presentation, and which not only talks about the technology, it talks about the perspectives, it talks about the various to address the need of the country or the region and redefine, rethink, and realign. I think we are living in the world where if we do not redefine and realign, we will be perished one day. So I think this is the time we need to broader level, then go into the specific. But I'm sure, with this audience in this discussion would come up with many questions and many queries. So before I ask any question or anything, maybe I'll open the floor.
Good afternoon, Dr. Hari, and of course, my other colleagues. Thank you very much for giving us your thoughts on this new technology. I have a couple of, I could just gather some points from what you said. What struck me most importantly is this 9.3 million liters wastage. Is a global number data or This is
It's a
recent data.
It's a global data, yes.
So is it because there is wasted, or it is a uncomponent separated type?
No.
I mean, it went down as no, it's not that, right?
In many countries,
it could
in
be
many
both.
countries, what happens is that you do get the plasma. Now, I know that at one stage in the Philippines, they were collecting hundreds of thousands of liters of plasma, but the plasma could never be sent to a fractionator to be fractionated. So what do you do with the plasma? You literally throw it down the drain, and 9.3 million is probably not a very bad estimate. In fact, I think it's a very conservative estimate, if you want to ask me.
So if Dr. Ranjit will let me ask a couple of questions more, because it's lying in a sequence, without breaking deal. And you said this technology being so simple, if it is a single capture step where you can actually capture all the proteins of your interest, have any fractionators bought this technology from you for them to do it, and at what scale does it work is one question. And if that is happening, then I mean, all the steps are down, the cost of production would have come down, should come down technically, which is what many of us are struggling with, because the cost of raw material is going more than the cost of final products. That's one. And the second question, a simpler solution when you think of out of the box, if it is a simple cartridge-like solution where you can actually filter out some of the small nano, that is the hepatitis B, C, A, D, I mean E, which is like lipid coated and non-lipid. Why did we not try it on as an inline kind of thing, during collection itself, so that you get a virus removed kind of a raw material to start with? So the starting material itself is of a better quality. If that is so, or is some buyer buying your technology and trying to do that, it should be good, because solvent detergent, I mean, there were so many other methods, and none of it actually reached the market at all. So at one time, in a country like India, or even say other countries in Southeast Asia, if this is doable, it would sort out a lot of things. So the next question that is coming up, based on what you were saying, is that if at all is it equivalent to a virus inactivation technology, or you need to incorporate virus inactivation technology yet again, because you're showing a good 6 log and a 7 log, etc. So do you still want, so if you combine two methods, you already have a 12 log algebraic sum. So are you going to claim that as your virus inactivation methodology, in which case you don't have to employ a solvent and a detergent and remove that, and sit and heat it or whatever? I mean, I'm just thinking aloud, okay? So these are some of the questions that I had for you. So is there somebody who has licensed this, and how successful is it if it was so simple? Because I remember meeting you many years ago, and I have heard of this technology that you were trying to talk to us about. This was many years ago, I think at the KEM hospital, or probably early part of my Reliance days. And simplified it and demonstrated to the world. Just curious to know.
The only thing that I'm very glad about is at least I've excited you about the technology. Look, let me try and answer your question. Firstly, we are a fractionator in our own right. So we use this technology internally. This is not a technology that we have licensed to any of the other established fractionators. So we have, as I said to you, we have fractionation plants here in Sydney and also in Brisbane. We will have one in Brisbane, and we're building one in Singapore. The technology can be used for anything as small as 30,000 liters per year, right up to 1 million liters or more, and which is what we are doing with the two plants. So here and in elsewhere in the world. So there is that ability to be flexible, which is some flexibility. The second part of your question, I believe, is inline viral reduction. You know, there are lots of things that you can actually introduce when you are doing things like this, and that is a possibility. But then you've got a very much a modular viral reduction step that could be introduced. Now, that is probably something that is worthwhile considering when you're looking at. In effect, you're not doing that at the collection stage, but you're
improving quality of raw materials. So
exactly. I understand where you're what you're saying, but this makes up for it. I have tried to make things really simple as far as the regulators are concerned, because when you have a new technology, the idea isn't to go in there and claim everything. So what we've done is we've said, this is your capture step, but we're still going to keep your pathogen reduction steps. But it comes back to the fundamental argument that I have here today, and that is that if you are looking at a final product that is ultra safe, when you cannot make, for whatever reasons, the sort of adjustments at the plasma collection level, then having to claim this as one of the steps for viral reduction ensures that the final product is in fact safe. So just so that you know, we are doing a lot of work so that we can go to the step in its own right, and it is. It's not only a viral reduction step. If I had to tell you what else we've done, it reduces endotoxins. We know that it removes prions. So it is in fact a total step in pathogen reduction in general. So that is something that we're very proud of, but it is definitely something that we want to keep hammering as we go on. I think I've answered all your questions. I mean, if I missed something, I apologize.
Thank you. Appreciate it. Thank you.
Thank you, Hari, for answering all the questions coming from Chandra. And I think Sangeeta has raised hand. Let's go to Sangeeta.
Thank you so much, Dr. Hari, for letting us know about this new technique. I just wanted to ask you one question. We are talking about, you said the quality of this raw material would be improved by it, isn't it?
Mm-hmm.
So the quality improvement is only on terms of the reduction of pathogens, not elsewise, isn't it?
Well, in terms of the product, the final product is IVIG or albumin. The quality of the product is still IVIG and albumin, and it has to meet with all the release specifications that we have when we are looking at that. So I'm looking at the safety part of it. So the quality of the product is always maintained. In fact, when you're using electroseparation, the actual biochemical characteristics of the molecule is conserved. For example, in IgG, you are conserving Ig3, which is in fact lost when you use cone fractionation. So the quality is always there. The quality is never compromised. The safety, that's your viral reduction and your pathogen reduction, is enhanced. And that is important to me, at least, and please, correct me if I'm wrong here, but it is very important. If you cannot have the screening that is required and the regulations and the plasma collection centers upgraded, then you've got to cross the gap somewhere. You've got to fill the gap somewhere, and that is what we've done.
Yeah, thank you. So what I understood was the quality of the plasma which is being collected has to be maintained as per the norms, and you will maintain it further by using this technique of means the pathogens and all would be reduced considerably more as compared to the other techniques.
That's right.
And I mean, you are also preserving the other factors
Yeah.
I just wanted to comment on this only, that we have to have the initial product as per the norms.
Oh, absolutely. And I'm not even
There is no
advocating
compromise
otherwise.
on that.
No, absolutely not. There's no compromise on that. The final product must never ever be compromised. There's no question about it, because as far as I'm concerned, it's going into a human being. It has to be the safest possible product available.
Yeah. Thank you so much. Thanks.
I thank I see Gopal has raised hand. Gopal, please go ahead.
Thank you, sir, for giving me opportunity to discuss something. I have some basic question from the Harish sir. Thank you, Harish sir, for giving lots of information and new technology introduction in this session. I have some basic questions that how many days it is taking to produce, taking one batch through this new technology, and what minimum scale we can start with this new technology. And third one is this reducing the BET level as well, apart from the viral reduction step? And what is the cost of this particular, if you are going to install this particular technology, is viable for the Indian market also?
Yeah. The minimum batch sizes, you can go down very low, but I would recommend that the minimum batch size is about 30,000 liters a year. So
30,000 liters per year.
Per year, yeah, exactly. So I think that's a batch size that I would definitely recommend. I think your second question was
how many days it is taking for one batch?
Okay, it's 1/5 the time that the current cone fractionation chromatography will take. So, I think right now you're looking at possibly about 5 days for the full process to be complete. When it looks at cone, we are looking at probably about 24 to 48 hours when you're using this technology. Right?
Okay.
Okay. And the third one was you were talking about
BET level, sir. BET level, bacterial endotoxin level.
Oh, right. The endotoxin levels are definitely reduced by this as well. There's no question about that. And the ability for the primary capture step to actually reduce the endotoxin is one of the advantages of the technology. Right. In terms of cost, it really depends on what we are looking at. A 100,000 liter facility obviously is going to be much cheaper than when you're looking at a 1 million liter facility. But let me give you a comparison. I mean, I think it's wrong for me to start telling you what the cost is, because we need to know exactly what it is we are doing. But let me give you a comparison. When Baxter built the 1 million liter facility in Georgia, in the United States, it cost them somewhere close to $1.5 billion. All right. Now, we're building a 1 million liter facility in Singapore, and the cost for the whole project is 400 million US. So literally, you're looking at about a quarter of what you would pay for a facility of the same size. So that is your cost comparison.
And this per liter, how much yield is there, sir? It's a double. Per
liter, the yield is 90%.
90%.
So let's talk about IgG. If you start off with 10 grams per liter or 10 milligrams per mil of IgG, you're looking at 9 milligrams per mil final yield, and that's it's a consistent sort of thing. And it doesn't matter which protein you're going for. The yield is in fact 90. Now we are very conservative when we say this. We usually say yields above 80, but we are looking at a 90% yield.
Means one per liter it is giving 10 gram or 9 gram.
If you start off with 10 grams of IgG, you end up with 9 grams. So it's 90%. Okay.
So, but sir, in this chromatographic technology also it is giving 10 gram, 10.5 gram.
Sure. If you use affinity chromatography, absolutely. But it's the cost. And affinity chromatography, to be scaled up to that level, is very expensive, and it depends very much on the resins that you use as well. So an affinity chromatography has got a mixture of cone fractionation and chromatography. It's not just one or the other. So your primary resource is still the cryoprecipitate. And in this case, it's directly plasma. We're not talking about anything you got to do. You take the plasma, you mix it with the buffer, and what you then get is the final product.
No, sir, we are using directly.
Yeah, but even then, it is expensive. It's affinity. So you and I can argue about this, but
Yes,
I've gone through this in many, many ways.
correct, sir. Correct, sir. Thank you, sir. But sir, one more question. This is single-use technology, means that cost you told me that this much of liter. This is for the single use. We can't means after that again we need to membrane.
The single-use technology.
Yes.
The cost is actually cheaper than what you would do using affinity. There's no question about it. The actual cartridges are much cheaper. There's no question about it. You're not going to be spending more because you're using single-use technology. And I dare say to you that the regulators actually like single-use technology because you're ensuring there's no cross-contamination.
Correct, sir. Sir, so we can buy from there to here to India. Now companies are buying, or just it is in Australia only?
This is our technology. This is AATM's technology. And if there's a project to be done in India, it's a project that we would want to get involved in. And you've got the person in India who is our representative, and that's Ranjit. So reach out to him and he'll be able to help you.
Okay. Thank you. Thank you very much, sir.
Ranjit sir, you are mute. Please unmute yourself. Ranjit sir, please unmute yourself.
Sorry. I'm sorry. Kalyan, please go ahead.
It's okay, sir. The floor is yours.
Do you have any
Yeah. No, sir. I don't have any question.
Any question, Kalyan, you were asking?
Okay. No. Thank you, sir. I'm just enjoying and learning a lot from you and Hari sir.
Thank you.
Ranjit,
Anybody has question from the floor? Please go ahead.
Yeah, Ranjit, there are, Dr. Prasad.
Dr. Prasad. Dr. Prasad, then Amjot. Yes, Dr. Prasad, please go ahead.
Okay, sir. Thank you, Dr. Nair, for your a nice presentation.
My pleasure.
Just I have a simple question, one-line question. So far as
no questions
safety is concerned, whether we can pyrogen. So far as pyrogen testing is concerned, we can test bacterial endotoxin test instead of doing the pyrogen test for a blood product.
Using a pyrogen, yes. Yes, you can. And that's something that we do normally anyway, because it's something that it has to be to meet regulatory specifications.
But so far I have the experience for that. In the case of plasma derived product, they have if they have the little bit of the like factor 8, factor 9, or anything, it or enhance the reaction of the bacterial endotoxin. So that's why I'm asking you whether it is feasible, because most of the pharmacopoeia they have advised you can do rabbit or pyrogen, rabbit or bacterial endotoxin test. But so far as my experience is concerned, bacterial endotoxin test is only feasible with the hemophilium, that is the product of the Takeda. And no other product is giving the good result. Either there is enhancement or the inhibition with that.
Well, we make it very, very clear that you got to have, there are obviously certain levels that are acceptable as far as our regulator, the Therapeutics and Goods Administration is concerned, and also what the FDA and EMA require. As far as all the products that we have manufactured, that's well below what the actual allowable limits are. And, that's something you've got to be very careful about. There's no question about it. But also that endotoxin pyrogen levels are very dependent on what you do when you are actually handling the product as well. So the cleanliness profile, the way that the machines are cleaned, all the protocols that are involved in it all contribute towards that. So there's no question about that. And that is one of the nice things about a modular technology, because if it's disposable, it means that you never have to worry about any contamination that may have been there because of a prior fractionation. You can actually throw that away and then start fresh with something that is sterile and clean.
And I have second question regarding the regulatory system in the Australia or other country. In India, we are doing testing as per our pharmacopoeia, Indian pharmacopoeia monograph.
Yeah.
But I have the experience with the other countries, they do only 5 or 6 parameters and release the product.
Mm-hmm.
And just I will give the one example. I got one product from the Germany that was released from the Paul Ehrlich, Germany, and that was failed on the basis of protein content. And when I contacted with Germany, they told me, Dr. Prasad, we are not doing protein content testing.
Is that right? Okay.
So that's why I ask each and every parameter is the important, whether it is the pH, content, sodium, potassium, whatever.
Yeah.
So we do all the.
Yeah. Now, the only thing I can tell you is that our TGA in Australia is very stringent with the number of facets that we have to go through before we have product release. So I'm not sure why they would not do protein testing in Germany, because they seem to be much more rigid over there anyway. So it'd be interesting to see, what product it is and why they would not want to do something like that.
A product by the albumin, but I cannot take the name of that company because
of course, appreciate that. Yeah,
confidential, and I was the regulator. Yeah, thank you, Dr.
Okay. Thank you, Hari and Dr. Prasad. Navjot, please go ahead.
Yeah, I have got one suggestion, and I might request because I have seen that there are members from the different countries. They're attending this webinar. The first, my suggestions will be, as Dr. Hari from AATM, we actually sincerely thank you or showed our gratitude that you have given a very, scholarly lecture. But, the content of the lecture, if it is not recorded and put in the YouTube, I think memory goes not beyond 72 hours. But my request is that because there are knowledgeable people in this particular webinar and in the field, and this is a long felt need, that what are regulatory process and how we should actually fit into the regulatory process by the developing countries, especially AATM countries, when they prepare, because you are right, absolutely, that lots of plasma is thrown away. I do not name the country, but I know at least around 8 or 10 countries doing good plasma, and it is not actually channelized. Therefore, my suggestion is that if we can, make a small, some sorts of, small resource group, which can actually develop some regulatory parameters for AATM document. This is a document. And this will be in our website, which will be helpful for our members as well as globally. So what are the regulatory things we should keep it in mind when we are talking about fractionation? The plasma we produce, vein to vein, as well as what are the parameters we should follow. That is number one. And if this everybody agrees, I request Kalyan and Sangeeta, we can pick up one from, of course, I know Dr. Laxman is here, I can see him. He can nominate somebody from Sri Lanka. Then I can see Dr. Rekha from, Nepal. They already were working on it. They came out with a document. I can see Dr. Nova, she's from Indonesia. They are also working on it. So it is multinational, some AATM member group, and with a time frame, not that it is going to linger and linger. And of course, Dr. Hari, Dr. Prasad, Dr. Sandra, Ranjit, all are there. So I leave it to Sangeeta as well as Kalyan to give something out, document really useful for the AATM. And that's the suggestion. And all this name I have just spelled out, if they have something to say. Over to you.
I think
Thank
that's
you.
actually
Thank you, Navjot. Maybe Hari can address it.
Yeah, I think that's a very good suggestion. I think that having a working group to actually look at that is the first step in trying to get a integrated approach to this problem. You have to start from somewhere, and I think your suggestion, Dr. Chaudhary, is perfect. It is exactly what I was trying to get at, where an association like AATM can take a front foot in getting something like this going. So by all means, I'm happy to participate and contribute. By all means, it make a huge difference as far as I'm concerned.
Thank you, Hari. Any questions from floor before I go forward? Okay, so I think if there's no question from the floor, let me spend a few minutes with Hari.
I have a question.
And maybe it will help. Yeah, please go ahead. Anybody has question? Okay, so Hari, I was just wondering.
Yes. Hello, everyone. This is
Hari. Yeah, please go ahead.
Yeah, I have a question. This is
Yeah,
Shekhar
please go
from
ahead.
Hyderabad. Yeah, thank you for this session, and I have 2-3 questions for Dr. Hari. First question is, with this Agros technology, how many molecules we can purify from the plasma? And my second question is, you mentioned like with Agros technology, you can get around 7 to 7.5 gram per liter of yield. So in that, what would be the IgA content, which is basically a contaminant in IVIG?
Yeah.
And if possible, please mention the yields of albumin and factor 9.
Yeah. Let me answer. Your first question was how many proteins you can pull out. You basically can pull out any protein that you want. It's just a question of targeting the protein and pulling it out. So there's no question about that. The second question you had was in terms of yield. It doesn't matter which product you're looking at, the yield is about 90%. So albumin in fact is even higher. We can pull out albumin at about 98% yield, and that's a very consistent sort of approach that we've got for a protein like albumin. And I think there was the third question was, I can't remember. Sorry, can I ask you to ask the third question?
I want to know what would be the IgA content in IVIG, which is purified by Agros technology.
There is no IgA contamination. In fact, we've been very, very careful in doing a mass spec analysis to see what the IgA contamination is. And in fact, of all the profiles, no, it's very, very much within regulatory requirements. In fact, as you know, IgA is a tough one. But we